Dr. Hood, I like your approach. It reminds me of an article I read, and I can't tell you exactly how many years ago because of the telescoping effect. Could have been 10 years ago, could have been 20 years ago, could have been 30 years ago. But there was an article written about sending this tumor to the lab and do essentially a sensitivity type of test as we do with antibiotics for bacteria. And so I think let's look at the glioblastoma question. Realizing that all the patients that you've had have a lot of noise in the system, meaning that there's a lot of variation within the tumor itself, but there may yet be some critical feature within the tumor. Now, have you or others the ability to culture the tumor and then look for commonality among the various tumors as a way of looking at potential therapies? I would like to think that we all have pretty much the same chromosomal numbers, and we probably all have fairly similar genetic complements, and what causes someone to develop a glioblastoma versus others within the same family or the same population not, might boil down to some spontaneous mutation within a subset of chromosomes or genes. Sure. That might lead to it. And I was just hoping that we might be able to culture these glioblastomas with the idea of finding a sensitive cell or at least a progenitor of the ultimate malignancy. Thoughts?
So yeah, I will say glioblastoma is a tumor that's among the easiest to culture cells for. So really good people can have very high rates in the probably 40 or 50%. It's not 100%, so there are some that don't work. I think the other important point is by sequencing each of the 3 tumors or 4 tumors and comparing it against the germline of the individual, you can actually identify a lot of the major mutations for that tumor that are the real driver mutations. And the first place you'd want to look for appropriate therapies is to see whether there were kinases or whatever that could map into those tumors. But it is possible to do the culturing, and in fact, I didn't talk about that. We are culturing these and doing a lot of single-cell analyses, and we're finding really interesting quantized populations in multiple tumors. And you wonder what those populations are and should one of them be the target for drug therapy in the future. So there are possibilities for doing that.
Okay. So, I'm wondering if you can say anything more about wellness proteins. Dr. Hood, you mentioned those a few times.
I should never have mentioned that.
Well, but that's probably a super important point.
So it is. It's really an interesting concept. So I would say one of our genome studies is looking at a series of individuals that are 115 or older, and families are tough, right? So although you can go back a generation or two and find families, and we're going to start doing that. But the interesting idea is, do those individuals show a purification of, quote, wellness genes that sets them off from their normal counterparts? And Eric Topol is doing a similar kind of thing. He has these people he denotes as the welderly. So these are people that are 90 years or older who've never been sick for one day in their life, and he has 1,000 of them. So he's sequencing their genomes, and it's exactly the same approach. It's, can you see an enrichment of, quote, wellness genes? Now, on the other side, what we really need are metrics for wellness, because we really, you don't want to just say, well, you're well. You'd like to be able to take measurements and say, are you getting weller, or are you staying the same, or are you going downhill? So Stuart Kim at Stanford has actually done some really interesting transcriptional studies on kidneys from all age ranges. And he found a profile of transcripts that seemed to predict the tendency for people to look physiologically much younger than their chronologic age was. And in a sense, that's a metric for wellness.
But you have to have their kidneys to do that?
Yeah. Well, so we're trying to see, we're just starting to try and look at whether we can do that with white blood cells. And we don't know the answer to that. Yeah, you don't want to do kidney biopsies for a routine wellness assay.
I have a question for Lee Hood as well, which is, to what extent are the current blood-based tests limited in terms of sensitivity and specificity by limitations in the platform, in terms of, what concentration of how abundant the protein has to be and the noise in the measurement and so on?
So for proteins, there really is a fair limitation because, as you know, the blood has a dynamic range of protein concentrations. It's about 10 to the 10th. And in fact, we couldn't even do the assays that I talked about unless we had columns that could remove most of the 15 or 16 most common proteins, which would be about 97% of the blood by mass, okay? And if you do that successfully, then that lets you drop down 2 logs of magnitude in the proteins you can detect. But if we could do that more effectively, we could do even much better. And that's why we've always started our screens with large numbers of proteins, because one of the criteria is going to be it has to be in that range where you can detect it, obviously.
I had a question for Mary. I was struck by that wonderful work that you did where you showed the IPS differentiation didn't finish or didn't make it full. Do you think if you took those human cells from the patient, did that to them, then put them in the mouse brain, and they're marked by the back tag system, that they would finish all of that and you could recover them again?
Well, that's exactly what we're looking at. I mean, we did do the one trivial experiment following up on Jim Darnell's work, 30 years ago, just to see that if we took the cells out of the brain and put them in a culture dish, do they automatically lose this? And they do not.
And they do not.
So there's a very linear path ahead of us to either co-culture the cells with their various synaptic partners and/or being able to put them back in the brain is a great thing. We haven't done it with the human cells, and it's been done a lot with glial cells. But it'll be important to know if you take a human cell whether you can assay migration, period, whether they'll work on the mouse glial process. I think they will, and also whether we can replace Purkinje cells because that's very involved in disease, of course. But the systems biology is going to get us there for sure.
And you seem to be edging into it.
Yeah, no, definitely. No, I'm a great believer, even though I didn't talk about it much.
For Dr. Hood, just to follow up to the sensitivity, how quickly do proteins in the blood come up as a readout for brain injury, and would you get more sensitivity out of CSF?
So the answer is we haven't done longitudinal studies at all on any of these, and that's really a critical kind of thing. And it's especially critical for early diagnosis. We really want to know what the sensitivity is going to be, so we can't answer that now. My feeling is that it could be well that cerebral spinal fluid would be a much more effective fluid to do the diagnostics in. It's just that it's not a routine procedure, and you're certainly not going to do routine scanning and things like that. See, I'm arguing, one of my thoughts in the future is, wouldn't it be wonderful if we could identify of the order of 50 organ-specific proteins from each of our 50 major organs, And wouldn't it be wonderful if we had microfluidic assays that could take a fraction of a droplet of blood and quantify those for each of us every, 6 months or a year. And then what you'd have is a readout on all your organs that A, established what a normal baseline was, but B, it would follow any transitions from that norm into, this something that's changed, and here are the set of categories that you ought to be worried about in that particular organ.
And save people a lot of treatments that are horrible.
It would save it. And you could do it with a finger prick, too, if you really can get a fraction of a droplet of blood. Yeah. The other thing I'm really interested in, but it turns out it's not trivial, is I think saliva could be an incredible diagnostic. But I'll tell you, the proteins that are in saliva are probably even worse than the dominant proteins in the blood. Worse because we don't have any antibodies for most of them, so we can't get rid of them easily. So they won't let you get down into those lower levels of interrogation. But we're going to work on that. So.
I was thinking with your TBI example, we have so little to offer mild to moderate TBI patients that if we had a diagnostic tool early on, then instead of just having a negative CT scan, sending them home, that intensive consulin or something might be possible to prevent a lot of the sequelae.
Well, I'll tell you, if we had that, I'll bet no parent would ever let their kids play Pop Warner football.
No.
And I would argue no parent should ever let their kids play high school or college football either. Now, there's a big argument about that, and the NFL side of the argument is that there are people that are genetically resistant to TBI, okay? And therefore, and their argument is, that's true of most of us, so we only have to worry about a small fraction of people. I don't believe that for a second. And there's no data that even speaks to that issue. So once we have an assay, we'll be able to sort that out very quickly.
I have a quick practical question for advice from any of you. Per IRB, we asked for blood. I get why we need that, but then the backup is saliva. We're finding boys 9 to 14 will spit, but they don't want to give a small blood sample. Have you observed this, and do you have any advice how to persuade them? The girls, it's not a problem.
Is that right? I find that interesting. But you know what I'm doing? We're having a big family retreat at a family ranch in Montana in the 1st of August, and we have 3 generations of hoods and relateds coming, and I've almost persuaded all of them to agree to give blood so we can have a complete genome sequence done on the entire family tree. And I have a few resistant ones, and after I'm finished with them, I'll have more experience with that. But it's not resistant. The resistance isn't based on giving blood. It's really based on much more legitimate questions of
whether they want to know.
Well, no, whether they want to know. We will not tell them anything they don't want to know. No, the real problem is, can your insurance company get access to this information? Oh. And see, there is a law that starts to deal with it, but it doesn't completely protect us. So it's a legitimate issue, I think.
I'm happy to talk with you about practical issues around this because we deal with it with autism and all these things all the time. So we can talk about it afterwards. There.
I know nothing about it.
Yeah.
This is why I collaborate with MDs.
Well, I have a project with the Pacific Science Center on head injuries. And my belief is, just as you promoted, that no kid should play contact sports. And there's actually a lot of evidence about brain metabolism and function immediately after any kind of head injury, and it goes on for not just hours or days, but weeks. And the problem with it is the parents are putting their kids at risk for a lifelong problem of survival and success in the world. And so I think I tried to get professional football to be outlawed, but you know what? I'm going nowhere with that.
Yeah, I can imagine.
But I think we do have some reasonable rules within the state of Washington that have looked critically at head injuries and have tried to keep kids out. But I think the bottom line is that no kid should play these kinds of sports. And that includes soccer, where heading the ball is part of the issue. So, I think that part of our problem is that we need to educate not just parents, but people. And the other question you asked about insurance, my bias about insurance is we don't need them because they don't provide any benefit to us in terms of our health care.
But the point I would make is, wouldn't it be wonderful to have a quantitative assay that could measure these things?
Absolutely.
That would be transformational. And see, with a quantitative assay, you could persuade parents.
Yeah.
You could say, look, this is your child's normal, and look what's happened here. And we can map this right into other people that have had TBI and follow the length of it and all of those kinds of things.
That requires a head injury.
There are a number of companies working on it.
Well, we have lots of people with head injuries. They're called soldiers. We have an enormous population there that we can look into. Yeah. And the other place that has PTSD, TBI are police cadets too. They especially PTSD there. There are
things they have to see.
Yeah, and I had a friend who said there are three kinds of police cadets. There are those that from day one are stressed and never, ever recover from it. And you ought to have an assay that says, you should never go into this. And there are others that have it and can go down quickly, or have it and very, very slowly go down. And even that third class is compromised functionally for what they have to do. So you'd really like to have tests that say, choose another profession. Yeah.
Any more questions? Then I have one brief question for Dr. Geschwin. You had this nice finding where you have multiple different mutations converge to one gene expression profile. Would that mean that these different mutations are not very useful to stratify diseases? And if we see that a lot in other diseases, it means that it's a challenge to use mutations to stratify disease, right? Because
no, I wouldn't say that at all. What we're looking at is a kind of pathology. And I think that actually what we call inverse mapping, or starting from these mutations that have large effect size, could give you a lot of information. For example, studying people with distinct mutations and the phenotypic heterogeneity would be really interesting. The question is, where is the convergence? The convergence we're seeing in 70% or so is kind of in brain system. Where you might want to intervene is in a very specific molecular signaling pathway, which would be quite a bit earlier. And it's likely that there are multiple signaling pathways that are converging on neuronal dysfunction and developmental abnormalities, et cetera. So I think instead of having 1,000 different diseases, we may end up with several dozens. But I think similar to cancer, we have to think of it as a whole bunch of rare diseases where we're going to be using molecular targeting. So actually knowing that could really help stratify patients. One last point that just highlights, I think, what Dr. Hood was talking about and really brings it home is that Stan Nelson at UCLA instituted exome sequencing about 1.5 years ago as a medical test in our institution. And so there are a lot of pediatric cases and neurologic cases that are now getting sequenced. And we're making a genetic diagnosis in almost half of them, and in about a quarter finding something very unexpected and relatively new, and about the other quarter, that other half of that half, something relatively expected. So that's just with an exome sequencing, and that's just with our current very rudimentary knowledge. So I'm really kind of pumped about precision medicine, or whatever we want to call it, going forward, and having genome knowledge.
Okay, any more questions? Any more final remarks, panel? So let's thank the speakers again and have a discussion.
I do have one announcement to make. We have posters on the 3rd and 4th floors to persuade you to go up there. We have really good appetizers and drinks, and you can go to the 3rd and 4th floor by elevators, which you can access in the main lobby.